Hello, my name is Sasha Ebrahimi. I'm a chemical engineering PhD candidate at Northwestern University, working with Professor Chad Merkin. I want to begin by saying that I'm looking forward to seeing all the talks in this student competition and biosensors session, all the really exciting and impactful work that people are doing. What I want to do today is tell you a little bit about the work that we've been doing towards creating tools that enable us to study the biology happening inside of living cells in a new and better way. But to do that, the first question I really need to answer for you is, why are we interested in looking at things inside of live cells? The simplest answer to that question is that the molecules inside of cells have a big influence on how cells behave. Every single disease can be associated with dysregulation of one or many molecules inside of cells. Now, because these molecules are dynamic, meaning their levels change with time, we want to be able to get dynamic information. We want to look at things in live cells. What I'm showing you on this slide is the chemical composition of cells: proteins, nucleic acids, so on and so forth. What I would say is that there are powerful, not perfect, but powerful techniques to study proteins inside of live cells. This is based on something called GFP and was a subject of a Nobel Prize in 2008. There are powerful, not perfect, but powerful techniques to study nucleic acids, for the most part in fixed or dead cells. These are techniques like FISH or PCR being the subject of another Nobel Prize. Then you have this whole left side of the circle that's collectively known as a dark matter of cells because there's such a lack of tools to be able to study them. So the point of the presentation I'm going to give today is to answer for you how it is that we would develop tools that not only enable us to study proteins and nucleic acids, but enable us to study the rest of the circle as well. To do that, the theme that we're going to come back to over and over again today is the use of probes made out of nucleic acids like DNA. Why is that? Well, DNA has a lot of advantageous properties. If I were to summarize the properties that I've highlighted on this slide in one sentence, what I would say is that DNA is a biocompatible material that I can synthesize at a relatively low cost, relatively quickly, with a desired sequence and a desired length. And by tuning that sequence and that length, I can program DNA to recognize and bind to different types of molecules. How? So in some cases, DNA can bind to its complementary strand. This is the classic A binds to T and G binds to C. In other cases, you can synthesize things called aptamers, strands of DNA that are able to adopt a conformation or shape in solution that allow them to bind to things like small molecules, ions, or proteins. It's very powerful to be able to use these type of structures to look at things in complex media like serum. But when it comes to using these things inside of live cells, there are a lot of challenges. Remember, DNA is negatively charged, the cell membrane is negatively charged. The question becomes, how do you get these things into cells? And oftentimes the answer is you need toxic chemical reagents. But even once they're in cells, there are these things called nucleases that like to chop up and degrade DNA. So for instance, if I have an aptamer inside of a cell and it gets degraded, it's not going to be able to bind to its target. I won't know that that target is there. To overcome these limitations, for the last 15 years, the Merkin lab has been working on a new class of structures called spherical nucleic acids, or SNAs. It turns out that if you take nucleic acids like DNA and densely functionalize them around a central nanoparticle core in what's called a spherical nucleic acid architecture, you start to get some really interesting properties. These structures actively enter cells. Once they're in cells, they're more resistant to nucleus degradation than their linear cousins. But from the standpoint of designing sensors, we know that the name of the game is sensitivity and specificity. It turns out that these structures actually bind their complements 2 orders of magnitude more than their linear cousins do. It also turns out that these structures exhibit cooperative and narrow melting transitions. What this means is that you can start to distinguish between targets that look a lot like one another, say targets that may have a single nucleotide mismatch. This speaks to specificity. But then the question becomes, how do you take one of these structures and make them into a probe? Now what we're going to do is start with a normal SNA, gold nanoparticle functionalized with DNA. But now let's assume that the DNA on the surface has 20 base pairs of complementarity to some mRNA we want to detect inside of a cell. So these are recognition strands that we've attached to the surface. The trick is that what we're going to do is hybridize to these recognition strands short what are called flare sequences. These have 6 or 7 base pairs of complementarity to the recognition strand and contain a dye. When dyes are in close proximity to gold, gold is a really good quencher of fluorescence. In other words, fluorescence is off. Now, when I introduce a structure into a cell, these structures are called nanoflares. What will happen? Well, the structure will encounter its target mRNA. The mRNA will come in and bind and displace the flare. Remember, the mRNA has 20 base pairs of complementarity to the recognition strand. The flare only has 6 or 7, so it's a numbers game. So what will happen is that the flare will be displaced. Dye and gold are no longer in close proximity, no more quenching, fluorescence is turned on. At the time that the nanoflare was introduced, it was actually the only way to measure the genetic content of live cells at single cell resolution. And the real beauty of this approach is that the recognition strands on the surface don't need to be complementary to some mRNA. They can be recognition strands that are aptamers that recognize small molecules, ions, or proteins. And because of these capabilities, the nanoflares enable a lot of very important studies and technological innovations. For instance, nanoflares have been used as the first platform capable of isolating circulating tumor cells from blood based on genetic signatures. They've also been used for high throughput drug screening and the detection of diseased tissues in vivo. So that's the good news. But just like with any first generation platform that enables new capabilities, it's equally important to ask the question, what are the challenges? What are the limitations? And how do we overcome those challenges and limitations? The limitation of nanoflares that I want to talk about today is the idea of false positive signal. What do we ideally want? We want our target to come in and bind, displace our flare sequence, separating the gold from the dye and turning off fluorescence. But there are other pathways that we can go down where this can happen even if our target of interest isn't present. What do I mean? Remember, before I said that SNAs are more resistant to nuclease degradation than their linear cousins, but they're not perfect. If they get degraded inside of cells, the gold and the dye will become separated from one another and you'll see fluorescence, even if your target of interest isn't present. The same thing can happen for flare dehybridization or destruction of the gold-thiol bond. So as an engineer, you really have two choices to how to fix this problem. You can fix each of these three problems individually or fix the unifying limitation as a whole. The unifying limitation as a whole is that the fluorescence is only off when the dye is in close proximity to gold. So if you're able to make fluorescence turn on dependent upon not proximity, but something else, you may be able to overcome this limitation. So how can we go about doing that? What we want to do is take inspiration from nature. On the very first slide of the presentation, I talked about GFP. And there I talked about GFP because it's really revolutionized biology from the standpoint of how people are able to image cells, tissues, and even whole organisms. But I'm bringing it up on this slide to ask a very important question. Why is it that GFP even fluoresces in the first place? So if you look at the crystal structure of GFP, what you'll find is there's this chemical group called HBI that runs through the middle of it. When HBI is allowed to rotate around this methane bridge, you don't see fluorescence. When HBI is restricted in its rotation around this methane bridge, you do see fluorescence. So that's why in an intact GFP molecule, when HBI is running through the middle of it, it's in a rigid environment, restricted in its rotation, you see fluorescence. When GFP is denatured or degraded, the dye is no longer in a rigid environment, rotation allowed, you don't see fluorescence. Now the next question becomes, what is the equivalent of this idea that we just talked about with proteins in DNA? And the answer to that question is DNA intercalators, dyes that when free in solution don't fluoresce, but when they intercalate between DNA base pairs, their rotation is restricted and you do see fluorescence. Now the big challenge with that is that these DNA intercalators are very nonspecific. In other words, they'll intercalate between any DNA base pairs. What do we want? We want our DNA intercalator to only turn on its fluorescence when some target of interest binds to our DNA and changes the DNA structure in some way. How do we go about doing that? That's the idea behind a platform that we developed here at Northwestern University called forced intercalation or FIT aptamers. How does it work? So you can imagine, if I take an aptamer, which I'm showing you here, what we're going to do is covalently conjugate our dye to that sequence. So that's what I'm showing you here as the tip of this pen. By covalently conjugating it, the intercalator is not free to go float around and intercalate into any DNA base pair. But in this conformation, the dye is free to rotate. But what happens when I add a target? Well, when I add a target, the aptamer will bind to it, and the aptamer structure will change in such a way where the dye will now be in a more restricted environment. And as such, its fluorescence will be turned on. And that's the idea behind FIT aptamers. What I'll say is that we were able to show that we can interface this FIT strategy with every aptamer target binding mode in the literature. In other words, presence of a target may lead to intramolecular conformational changes in aptamers. It may lead to 2-epitope binding, or it may lead to split aptamer binding. And across all of these different contexts, we're able to show that we can detect clinically relevant targets, markers of cancer, stress, thrombosis, and heavy metal poisoning. But for the experts in the crowd, they may be asking, while it's powerful to be able to use these probes in different contexts for important analytes, do you have any advantages over state-of-the-art platform? And we're really excited to say that yes, we do. So we observe up to 20-fold enhancement upon target binding, and these probes are resistant to false positive signal. But we also observe higher signal-to-noise ratio than FRET-based systems. Why is that? So if you take a look at the aptamer I showed you before, and I'm holding here, you can imagine that you could put a FRET pair on both ends, a donor and acceptor, such that when a target comes in and binds, that FRET pair will come close together and you should see a FRET signal. But it turns out DNA is not really a straight line like I'm showing you here. It actually has a small persistence length, or in other words, it's very floppy. So at any given time, those two ends are very close to one another, meaning there's a high background signal, meaning that there's a smaller fluorescence enhancement. You also find that these FIT aptamers are kinetically superior to systems requiring strand displacement. This is intuitive. So if you take a look at this structure down here, which is a nanoflare, you need 2 events to happen before you see fluorescence turn on. You need your target to bind to the aptamer, and then you need your strand to get displaced. Two events, meaning it's slower. With a fit aptamer, you only need one event to happen, binding to your aptamer. So if you put all of this together, you find that fit aptamers are actually the only light up aptamer that can detect a wide variety of targets with only a single modification. Now we have a quencher-free approach. Fluorescence is no longer dependent upon proximity to something, so we can test some of the claims that I made before and ask the question, can we make a new class of false positive resistant probes for making measurements inside of live cells? So what we're going to do is we're going to take the fit aptamers I discussed on the previous slide, and we're going to construct them into SNAs. So this nucleic acid shell that you see here are fit aptamers. They can detect analytes, and they facilitate cellular uptake when they're put on a nanoparticle in dense functionalized form. But the real beauty of this approach is that we no longer need a gold core. Remember, we needed a gold core before because it needed to quench fluorescence. We no longer need that. We can pick any nanoparticle we want. So what we're going to do is we're going to use a protein nanoparticle core. Why? Well, it's biodegradable, biocompatible. We know that proteins have several chemical handles on the surface that allow for us to functionalize things to them, such as nucleic acids. And down the line, we may even imagine using proteins that can detect something themselves. As a model target of biological significance, we'll talk about pH, implicated in various cancers and neurological disorders. It turns out that certain sequences of cytosine-rich DNA are able to adopt this tetraplex-like structure at low pH called the I motif due to hydrogen ions bridging cytosines into cytosine-cytosine-like base pairs. So you can imagine, in this conformation, if D is the dye is free to rotate. When pH is lowered, the structural change causes the dye to be in a more rigid environment, and fluorescence is turned on. So if we take these fit aptamers and we put them on a protein core, we're going to use beta-galactosidase as our protein core, and then do the experiment in buffer to see how these respond to difference in pH, we get a very nice up to 10-fold fluorescence enhancement as we lower pH. And if we just do a control where we put T-rich sequences on the surface of our protein, in other words, sequences that will not form the I motif, we see no change in fluorescence. So now, can we actually look at things inside of live cells? The answer is yes. So if we take one set of cells and expose them to pH 5.5 clamping buffer equilibrates the pH outside of the cell with the pH inside of the cell, and take another set of cells and expose them to pH 7.5 clamping buffer, and then analyze the fluorescence of these two cells using flow cytometry, what do we find? We find that cells treated with our I motif SNAs at pH 5.5 exhibit roughly twofold enhancement in fluorescence compared to those cells at pH 7.5, telling us that these probes actually are able to function inside of live cells. Now we can start to think about pushing things one step further. On the previous slide, we used beta-galactosidase as the core. But earlier I hinted at the fact that we can use any protein core we want, meaning that we can use a functional protein core. That's exactly what we're going to do for the next few slides. Using glucose oxidase, which from the name you can imagine is a protein that specifically recognizes glucose to make measurements inside of live cells. And to do that, we're going to need to develop a two-step assay. So how is this going to work? In the presence of glucose and oxygen, glucose oxidase catalyzes the formation of gluconic acid and hydrogen peroxide. So if I add this dye called FBBBE, which specifically recognizes hydrogen peroxide, I now have an assay for glucose. But the trick is that this dye is cell permeable when these boronic esters are here. So when I add it to cells, it enters in. But when these boronic esters are cleaved in the presence of hydrogen peroxide, this dye becomes negatively charged. So it can't just turn around and come back out of the cell. So this is how we are able to actually make the measurements inside of live cells. We can test whether these actually work. If we take a mixture of our glucose oxidase SNAs and hydrogen peroxide sensitive dye and titrate in more and more glucose, we see we get fluorescence enhancement up to 120-fold at physiologically relevant concentrations of glucose. Now, one of the big advantages of using a protein is that they've often been evolved by nature to be highly specific. And that's exactly what we see here. We only see turn on the presence of glucose and not in the presence of other off-target sugars. But now can we answer the million-dollar question? Do these actually work inside of live cells? So let me walk you through each of these three panels. To one set of the cells, we do nothing, and we run flow cytometry on them. So that gives us our baseline reading. To another set of the cells, we just treat them with our hydrogen peroxide sensitive dye. And we see a slight increase, which means that there's some basal level of H2O2 inside of cells. But what's really exciting is that we see a big fluorescence turn on when we treat cells with both glucose oxidase SNAs and our H2O2 sensitive dye, telling us that we are actually sensing glucose inside of living cells. I really hope that I've convinced you of today that we've come a long way in developing a multifunctional platform that allows us to make measurements using either protein-based or DNA-based recognition in high throughput at single cell resolution with high sensitivity and specificity. And the next question to ask are, what are we going to do from here? So the questions we're asking in lab now are, can we multiplex? Can we study dynamic processes? Can we track analytes? Can we look at low abundance targets, meaning can we introduce amplification strategies to our sensors? Can we push forward from glucose to look at other protein-based intracellular assays? And lastly, can we move away from semi-quantitative analysis to absolute quantification, being able to say there's, for instance, 10 millimolar glucose in this cell, 5 millimolar glucose in this cell. I want to end by thanking all the fantastic people that have made this work possible, in particular my advisor, Professor Chad Merkin, who has allowed us to pursue these really interesting research directions. I want to thank our funding sources. I want to thank you for listening, and I'm looking forward to any questions that you may have.